Hello everyone, today we're doing the Unit 5 of the International A-Level Biology Revision. Here are all the topics included, starting with Respiration 7a. Here are all the syllabus requirements. So respiration is a pretty hard topic to understand because it's just all over the place, but I'll just start really simply, and I'll link a few helpful videos down below. So first off, what is respiration? In the syllabus, it says that the overall reaction of respiration is the splitting of a respiratory substrate to release carbon dioxide as a waste product and reuniting hydrogen with atmospheric oxygen with the release of large amounts of energy. This just sounds really complicated, but let's break it down. So first off, the respiratory substrate is most of the time glucose, but it can also be a lipid, and it can be a protein too. This is just the basic equation from IGCSEs. And then we release carbon dioxide as a waste product, and we mix hydrogen with atmospheric oxygen to make water, and we release a large amount of energy. So what is this energy? This energy is actually a molecule named ATP. ATP is adenine triose phosphate. How is this energy released? So an ATP molecule looks like this. It has 3 phosphates, that's why it's called a triose phosphate. And then whenever we need energy for anything like active transport, for movement, or for making a complex molecule, we are going to break up those 3 phosphates into 2 phosphates in the diose phosphate, diphosphate, signaling for 2. And we're going to have 1 additional PI. It's PI because the I stands for inorganic. So the energy we get from respiration, but it's not actually from respiration. It's from hydrolyzing the product of respiration, which is ATP. The main purpose of respiration is to make ATP, so we can get energy from hydrolyzing it. Hydrolyzing it is just by adding water, and then we have ADP plus PI. So this ATP is then reformed from this ADP and PI by something we call phosphorylation. It makes sense because we're adding an additional phosphate to it again after it lost that phosphate in hydrolysis. This phosphorylation, where we add a PI to the ADP to make ATP, once again, the main point of respiration is to make ATP. It's either substrate level in the cytoplasm, or it's oxidative in a mitochondria. In aerobic respiration, both of these happen. But in anaerobic respiration, substrate level only. Substrate level produces much less ATP, but you don't need mitochondria and you don't need oxygen. So our benefits: we don't need mitochondria, we don't need oxygen. But here we need oxygen, but we make more ATP, and this has to happen in the mitochondria. Now the actual steps of aerobic respiration. If you feel thick in the head while listening to this, it's fine. You'll get through it. It's gonna take a bit, and if it doesn't, then I guess you're all just smarter than me. But you can do this. Okay, first we start with glycolysis. Glycolysis starts with the glucose. As you can see here, this is the complete equation. Here's the glucose we start with. We get it from the food we eat. So first we activate the glucose. What is glucose activation? We add a phosphate to it. The phosphate, first off, it prevents it from leaving the cell because it can't leave the cell membrane as easily, and it also makes it more reactive. It makes it more unstable. And then that phosphorylated glucose that we added a phosphate to, it's now called glucose phosphate. It splits into two, and then it's converted to pyruvate. While it's converted to pyruvate, ADP is converted to ATP, and NAD is converted to reduced NAD. What does this mean? Okay, so when NAD is a carrier molecule. It carries hydrogen ions and electrons to the last step, which is oxidative phosphorylation. Okay, so now we have a net yield. What we gained from this step basically is 2 ATP, and we have 2 pyruvate. You have to memorize the numbers, by the way. They can ask you, and they do ask it multiple choice. And then we have 2 NADH. NADH is the same as reduced NAD. Now we got a net yield of 2 ATP, 2 pyruvate, and 2 NADH in the cytoplasm of the cell. Now we're going to move on to the mitochondria. This here is a link reaction. It's the second step of aerobic respiration. It happens in the mitochondrial matrix. The mitochondrial matrix is a sort of cytoplasm, but for the mitochondria. Mainly what we're doing is getting this pyruvate that we formed here and turning it into acetyl-CoA. This acetyl-CoA is gonna then enter the Krebs cycle. The Krebs cycle is a series of reactions when the acetyl-CoA undergoes to produce ATP, NADH, and FADH. The NADH is produced from NAD and FADH is produced from FAD. And we also produce 4 carbon dioxides, 2 ATP, 6 NADH, and 2 FADH, and 4 CO2. This is from 1 glucose, and 1 glucose is for 2 cycles. 2 cycles. Okay, this is definitely not coherent at this point. But anyways, moving on. So all this NADH, or reduced NAD, that we've been making from the beginning, it's all for the last step, the oxidative phosphorylation. This is the one that happens in the inner membrane, or the cristae of the mitochondria. That's why we have foldings. We want a larger surface area in order for this to happen. This is the most important step and the one they ask about most. We already mentioned how reduced NAD and reduced FAD are hydrogen carriers. They're carrying hydrogen protons and they're carrying electrons. So here in the electron transport chain, you can see in the diagram, they release those electrons. Those electrons are passed along the electron transport chain from a carrier to carrier. Those are the carriers I'm talking about. They move along those carriers. This gives energy, or releases energy, to pump the hydrogen protons into the intermembranal space. What is the intermembranal space? It's the one you can see here. It's between the inner membrane and the outer membrane. So they're kind of just stuck here. When they're stuck here, it creates a proton gradient, or an electrochemical gradient, because there are way more hydrogens here than outside now, because using the energy from the electrons, we pumped all of them inside. Well, not all of them, but most of them. And this, as a result, produces an electrochemical gradient. Electrochemical. Okay, there's no space to write, so. Okay, after that, this is an ATP synthase. The ATP synthase is a channel where the hydrogen protons can pass through. When these hydrogen protons pass through, it spins. This spinning creates energy. This energy is used to phosphorylate the ADP. As we said, the main purpose of respiration is to phosphorylate ADP to make ATP. ATP is the one with the 3 phosphates, triose phosphate, so that you can use it and hydrolyze it later. So here, the ADP is phosphorylated using chemiosmosis. Chemiosmosis is when the ATP synthase has a proton gradient and the spinning motion gives energy to put an additional pi into ADP. Glucose from our food. We add a phosphate to it to make it more reactive so that we can split it into two. We split it into two and then we made it into pyruvate. In this process of making it into pyruvate, we used 2 ATPs and we made 2 ATPs. So we have a net yield of 2 ATP. And then we also made 1 reduced NAD, 2 NADH. And we also have 2 pyruvate. We're done. Okay, these two pyruvates, they go to the link reaction. This was in the cytoplasm. Now we are in the mitochondrial matrix, in the cytoplasm of the mitochondria, kind of. This pyruvate, it's turned into acetyl-CoA. This acetyl-CoA goes into a series of reaction called the Krebs cycle. This Krebs cycle, it makes more NADH and it makes FADH. And it makes ATPs. Where is this happening? This is happening again in the mitochondrial matrix, which is the cytoplasm of the mitochondria. And lastly, the most important step in aerobic respiration is oxidative phosphorylation. It's called oxidative because it uses oxygen. It's phosphorylation because we add a phosphate to the ADP. How do we add a phosphate to the ADP? So first, the reduced NAD and the reduced FAD. They release their hydrogen protons and they release their electrons. Those electrons get passed on in the electron transport chain from a carrier to a carrier. This releases energy so that we can pump the protons into the intermembranous space, this place right here between the inner membrane and outer membrane. So they're just stuck inside. This creates a proton gradient because we have way more here than here. Okay, and then using this electrochemical gradient, chemiosmosis happens. Chemiosmosis is when it goes out from here and this spins the ATP synthase, and then ADP is turned into ATP by adding a phosphate molecule. I really hope this made sense. Now, this is my summary. It's not very clear, but I like how I put everything, so I added it here. And anaerobic respiration. This, it also has glycolysis as the first step in the cytoplasm, but because we don't have oxygen, we do not have oxidative phosphorylation. So, our anaerobic respiration is different in animals and plants. In animals, it's called lactic acid fermentation, where glucose is converted to pyruvate, and then it's converted to lactic acid. This lactic acid, it can be converted into glycogen in the liver, or it can be oxidized further to release even more energy. But in plants and fungi and microorganisms, it's called ethanol fermentation. Ethanol fermentation is where pyruvate is added to reduce NAD or NADH, whatever you want to call it, and then that produces ethanol, carbon dioxide, and oxidized NAD. And here I show you each little circle. Most of the time I put it as a orange circle. This represents a carbon, acetyl-CoA. It has 2. Here a 6 carbon molecule, 6 dots, and I hope this makes everything clear. Here I wrote it down in case my handwriting isn't clear enough. And you have to know the equation for respiratory quotient. It's the CO2 produced divided by the O2 used. Okay, now that we discussed the actual respiration steps and how it happens, we're gonna try to measure the rate of respiration. It's measured using a device called a respirometer. This right here is a respirometer. So it's set up as shown in the diagram. Please memorize how to draw this. Most of the time you just have a drop of liquid in the tube, but the person here used a manometer. I don't know much about a manometer, but basically what we do is we put a sort of substance to absorb carbon dioxide. In this case, we put potassium hydroxide. You can also put soda lime, which absorbs carbon dioxide. So, the carbon dioxide that they let out doesn't actually have an effect on the experiment anymore because it's absorbed. So, the only change in volume of gas in the test tube is the oxygen that's coming in, it's taken in by them, so there is a reduction in the gas volume in the test tube. So, the colored drop or the liquid should come closer to the test tube. And then what we do is just basically measure the distance moved by the colored drop. And then we also calculate the rate, because rate is the speed of something. We calculate the time taken for the drop to move, and there we have it. We divide the distance moved by the time taken for the distance. Here we have common or just past paper questions about the first topic, respiration. So the role of the Krebs cycle. The Krebs cycle produces, produces NADH and FADH, which transports electrons for the electron transport chain in oxidative phosphorylation. It also produces ATP by substrate level phosphorylation. Another question, describe anaerobic respiration. This was for 5 marks. So the first step of anaerobic respiration is glycolysis in the cytoplasm, and then lactic acid fermentation, where pyruvate is turned into lactic acid and NAD is regenerated. The role of oxygen, this is by far the most common question. I see this in almost every past paper. The role of oxygen in ATP production. It's needed for oxidative phosphorylation. Oxygen acts as a final electron acceptor from the electron transport chain. It accepts a hydrogen ion from the reduced NAD or reduced FAD to form water. This is very important to memorize. We have the role of chemiosmosis in making ATP. This was for 5 marks, and I wrote down the answer. And finally, how is ATP synthesized by oxidative phosphorylation? It's synthesized by chemiosmosis. So electrons pass on from electron carriers, releasing energy used to move protons, and everything we discussed earlier. And we are done with 7A. Now starting with 7B, muscles. The first 3 points are about muscles, then we have 2 points about the heart, and the last point is about adrenaline. So I compiled some past paper questions here to summarize the first point. The role of tendons. Tendons are inelastic tissues that connect muscle to bone so that the bone can move when muscles contract or transmit the force. I'll get a picture really quick. The diagram here shows a joint. A joint is a point where 2 bones meet. The main question it asks about this is the role of a joint, the role of a ligament, and how movement is brought about. So it can show you a picture of someone either lifting a sort of weight or lowering their lower arm. Well, not like that, but anyways, okay, and here's the hand. And it'll ask you how. So you have to describe the flexor and the extensor. The flexor is always the muscle that decreases the angle between the bones. Here. This flexor, the bicep, it brings the bones closer together, so it's a flexor. While the tricep is the extensor. So you have to say they work antagonistically, which means they work in the opposite directions. When one contracts, the other relaxes. And then the bicep pulls on the radius, and the tendon transmits the force of the bicep onto the radius and pulls the lower arm upwards. The role of a ligament is attaching a bone to bone. It needs to be elastic so that the bones can actually move, while also maintaining the bones in place and keeping the joint stable. The role of a tendon is attaching muscle to bone. Here you can see a tendon, it attaches this ulnar bone to the tricep. The role is to transmit the force of the muscle onto the bone, so it has to be inelastic. Because imagine you're trying to pull up a book or something, and you use something that's really stretchy, a really stretchy rubber band, or even a piece of gum. The book won't be lifted because it just keeps stretching and it'll be really hard to lift it, so it has to be inelastic. Now the second point for this lesson in the syllabus is knowing the structure of a mammalian skeletal muscle fiber. Here I have diagrams to show you, but I really recommend you go watch some sort of animation to show you the sliding filament theory. I'll link a few below. So first, here we have the bone and the tendon, and we start with the muscle. A recent past paper question in 2021 asked to describe the structure of a skeletal muscle fiber, and the answer was a single cell that is multinuclear. It has actin and myosin bands that alternate to form a sarcomere. So as you can see here, a muscle fiber is a single cell, but it has multiple nuclei. So here's a single muscle fiber, and then here are 3 muscle fibers, and they're joined together and have mitochondria to provide the ATP beside them. Here is also a single muscle fiber, and it's showing you the filaments that it has. So it has actin filaments and myosin filaments. The thin actin filaments right here and the thick myosin filaments. When the muscle contracts, next slide, the sliding filament theory. So it's a theory because we're not really sure how the muscle actually contracts, but we have scientific evidence that this might be true. So first, calcium is released from the sarcoplasmic reticulum, the one you can see here, sarcomella or sarcoplasmic reticulum, and then it binds to troponin, changing the troponin's shape. I need to get a better picture, hold on. Here we have the sliding filament theory, or how a muscle contracts. Our main aim here is to shorten the sarcomere. The sarcomere is a Z-line to a Z-line, as you can see here on the muscle fiber, Z-line to Z-line. Here we have the actin and myosin right here. The actin is the thin one, the myosin is the thick one. Okay, so first off, what starts all of this is a release of calcium. So first off, calcium is released. This calcium binds to troponin. Troponin is this little molecule here. This troponin on the actin is bound to tropomyosin. Tropomyosin is the line here that's moving all along the actin. When the calcium binds to troponin, it pulls the tropomyosin away from the myosin binding sites. Here on the actin, we have binding sites that the myosin can bind to, but when the muscle is relaxed, it's covered by tropomyosin, so the myosin can't really bind to it. But when calcium binds to troponin and it pulls the tropomyosin away from the binding site, myosin actually binds to it, and this forms an actin-myosin cross-bridge. So this is the second step, a cross-bridge is formed. And then this creates some sort of tension that pulls the actin so that we can shorten the sarcomere, as we can see here. So the sarcomere is shortened as actin is pulled by the myosin here. And in this process, an ADP is lost from the myosin head. And finally, the myosin deattaches from the, myosin deattaches from the actin when a new ATP is bound to it. This new ATP causes the myosin to slightly change shape. The slight change in shape allows it to pull out from the actin-myosin cross-bridge. Here's a simplified diagram of everything. First, it's deattached and it has the new ATP, and then the ATP is hydrolyzed into ADP and PI, and then a cross bridge, actin-myosin cross bridge is formed, and the myosin pulls the actin so that the sarcomere can shorten and the muscle can contract. The power stroke. And one of the most common questions, if not the most common, for this lesson is the difference between fast twitch and slow twitch fibers. Slow twitch fibers are the ones that fatigue slowly. If you're going on a walk, you're using slow twitch fibers. But if you want to lift a heavy weight, or you want to go on a quick sprint that lasts 5 minutes, a quick race, you're using those fast muscle fibers. The fast muscle fibers mainly respire anaerobically, so they have fewer capillaries because they have less need for oxygen and glucose because they're respiring anaerobically. They don't need as much, and they have fewer and smaller mitochondria. They have large amounts of glycogen. They have the fastest rate of ATP hydrolysis in myosin heads, and they fatigue rapidly because they form more lactate, while slow muscle fibers have denser network of capillaries. They have a long contraction relaxation cycle. They have many mitochondria. ATP is supplied often from aerobic respiration, and they have small amounts of glycogen, and they fatigue more slowly because they have less lactate formation. In November 21, they asked us to explain how features of a slow twitch fiber are useful to marathon runners. Marathon runners run very long distances. I don't remember the exact distance, but it was like 21 miles or something. So they need every slow twitch fiber out there. So it has many mitochondria, which allows for aerobic respiration. It has a lot of myoglobin. Myoglobin is the pigment with a high oxygen affinity. It acts as a sort of oxygen store in muscles. You'll find that slow twitch muscles are more red in appearance because they have more capillaries, while the fast twitch fibers are more white in appearance. They have large capillary networks, so they have a good oxygen supply for aerobic respiration. Now moving on to the points about the cardiac muscle. First off, it's telling us to know that the cardiac muscle is myogenic, which means the depolarization is initiated in the heart. It doesn't need any external impulses to beat. So if you take out a person's heart, it will still continue to beat until it runs out of oxygen and the heart muscles die. It does not need any sort of external influence to keep on contracting. And then you have to know how the normal electrical activity of the heart is coordinated. I have a diagram here showing the heart. So first we have the SAN, sinoatrial node, and the AVN, atrioventricular node, bundle of His and Purkinje fibers. So the first initiation, depolarization, wave of depolarization, is initiated here in the SAN, and then this causes the atria to contract. And after a short delay, the initiation goes over to the AVN, which causes the ventricles to contract. And then it travels all along the heart. There is a slight delay, and then the ventricles contract. A question here from October 19 for 6 marks. It asks us to describe how the heart contraction is coordinated. So first we have to mention the fact that the cardiac muscle is myogenic and the stimulation for contraction is generated in the muscle. And electrical impulses are received from the cardiovascular center not to cause the actual heartbeat but to regulate the SAN rhythm. So the cardiovascular center can signal for the heart to beat faster. So the wave of depolarization from the SAN is sent through the walls of the atria, which causes atrial systole. And then the wave of depolarization passes to the AVN down Purkinje fibers after a delay causes a ventricular systole. If it asks you to calculate the cardiac output and it gives you the heart rate and stroke volume, you just multiply them. And you can use a triangle if it asks you for anything else. So cardiac output would be up, and then we have the heart rate times the stroke volume right here. Now starting with the ECG. So a recent question asked to define an ECG, and what you have to say is that an ECG is a graph that shows the heart rate or the heart rhythm, because this wave or this PQRST wave are the waves of electrical activity of the heart over a period of time. You have to memorize what P is, what QRS is, and what T is. P is atrial systole when the atria contract. QRS is the ventricular systole when the ventricles contract, and T is diastole when the ventricles are relaxed. So it might give you two ECGs, one of a normal heartbeat and one of a certain condition or a certain other person. And you can state things like irregular PQRST wave, or there is just no T wave, for example, smaller T wave. That's why you need to memorize what each one stands for. And you can also talk about the frequency. If one is a fast heartbeat and one is a slow heartbeat, you can say that the peaks are less frequent, or heartbeats are less frequent in the slow heartbeat. Why should the drugs that improve athlete performance be banned? Because they give an unfair advantage to the athletes that are using them. And then describe how the heart rate is controlled in response to changes in the blood during exercise. So during exercise, there is increased muscle contraction. Increased muscle contraction means we need an increased respiration rate so that we can make more ATP. Okay, so this increase in CO2 and lactate is detected by the chemoreceptors in the aorta and the carotid artery, which detect a drop in the blood pH. And then impulses are sent to the medulla in the brain, which has the cardiovascular center, or the CVC. And then impulses are sent by the sympathetic nervous system using noradrenaline to the SAN in the heart. And this increases the frequency of depolarization waves so that the heart rate increases, so that we can supply the muscles with more oxygen and we can again reverse the drop in pH of the blood. Describe the role of the SAN. It acts as a pacemaker for rhythmic contraction, and it initiates the depolarization to initiate the heartbeat. And you know how here we said the sympathetic nervous system using noradrenaline. Following exercise, we want to again reduce the heartbeat that we just raised, so we send impulses by the parasympathetic nervous system using acetylcholine. Now, as we know, exercise not only increases the heartbeat, but it also affects the breathing rate. Here, a question from January 14 for 5 marks asked how exercise affects the breathing rate. So the concentration of carbon dioxide in the blood increases, so the pH of the blood decreases. This is detected by the chemoreceptors in the aorta and the carotid artery. But this time, impulses are sent to the medulla respiratory control center. And then that sends more impulses to the intercostal muscles and the diaphragm to increase breathing rate. They increase breathing rate by contracting more frequently and more forcefully. Now, another question: why does a fit person recover from exercise more quickly? If you exercise more, then your lung capacity will increase and your cardiac muscles will get stronger, so you have an increased cardiac output. And you have more effective gas exchange. I'm sorry, this is a typo. And oxygen transport to muscle, so there is a more rapid reduction of oxygen depth. The role of adrenaline, this is the last point in our lesson today in the syllabus, the role of adrenaline in fight or flight responses. The syllabus does not need this much detail, but I hope it gives you a sort of overview or idea what's going on. So the sympathetic nerves, fight or flight. So it just enables you to either fight a sort of predator you encountered or run away from it. So all these things here, they help you. It inhibits bladder contraction because you do not want to be peeing when you're fighting someone. And the most important part in our syllabus is that the sympathetic nerves stimulate the adrenal gland to produce more adrenaline, because that adrenaline is gonna cause all of those. It's gonna help convert glycogen to glucose, so more glucose is in your bloodstream, so it can be readily used by muscles to fight or flight. And it accelerates heart rate. This is needed in our syllabus. This is very important. Accelerates the heart rate so that the muscles are also more oxygen is pumped to them, and you can fight off your predator. In the past paper questions, the students were asked, they were given this grid, and it had all the numbers of the heart flow normally and the heart flow when the person is stressed or when there is adrenaline. And it asked you why the blood flow to their brain remains constant, whether it's in a state of stress or relaxation. It's because brain is one of the most important organs in the body, and it needs a constant blood supply in order to function properly. And when you are stressed, the blood vessels to less important organs like the digestive system and skin constrict so that more blood can be delivered to organs that will be involved in the fight or flight, such as the muscles. Okay, so what does this adrenaline do in detail? Neurons of the sympathetic neuron stimulate the adrenal gland, and then increase impulses sent to the SAN, which increases the heart rate, and more blood is supplied to muscle cells. This stimulates the cardiovascular control center in the medulla oblongata. It increases the impulses traveling along the sympathetic neurons, affecting the heart, which further speeds up the heart rate. And also pupils dilate, the blood glucose increases, and the breathing rate also increases. Quick multiple choice question. The part of the brain involved in controlling the heart rate is the medulla oblongata, and changes in the blood pH detected by chemoreceptors also in the medulla. They are, yes, in the aorta and the carotid artery, but some are also found in the medulla oblongata. In the syllabus, it says that the changes in breathing and in heart rate are to deliver more oxygen and remove CO2 more quickly or more efficiently. Increasing the altitude, it will mostly show you a graph and show you that oxygen levels are lower at higher altitudes, and it will tell you why increasing altitude affects breathing. So in the graph, it showed that as altitude increased, breathing rate increased, because at higher altitudes there is lower availability of oxygen, so there is more anaerobic respiration and lactic acid is produced, and increase in CO2, so decrease in pH, stimulates the respiratory center in the medulla. It sends more impulses to intercostal muscles and diaphragm, contract more frequently and strongly. And it also affects the heart rate. Why? Because there is again lower oxygen levels, oxygen concentration of the blood will fall, detected by the cardiac center in the medulla, increase in heart rate to provide tissues with sufficient oxygen. Lastly, in 7b is a spirometer and a spirometer trace. So please memorize this. You have to know what tidal volume is, what residual volume is, and what the total lung capacity is, and get it from a graph in the vital capacity. The tidal volume is just how much air you're breathing in one normal breath. The vital capacity is in one sharp, forceful breath. So here it asks you which one here in the past paper question shows the tidal volume. So it's J, because here it's a larger peak, so this is a forceful inhale and exhale, while this is a normal breath. Explain how you would use this spirometer trace to find the breathing rate of a person. So you count the number of peaks and then measure the distance on the trace and convert it to time. And with this, we are done with 7b. In 7c homeostasis, we're going to discuss osmoregulation and thermoregulation, in addition to epigenetic modification from Unit 2 with the addition of peptide and steroid hormones. First, we have a question from January 18. Explain what is meant by the term negative feedback. Negative feedback is a certain mechanism that returns a change in a variable away from the normal value back to the normal value. This keeps a constant value or a narrow range of values in the body. For example, temperature or the blood glucose in the blood. Say the meaning of the term homeostasis. Homeostasis is maintaining a steady internal environment or body conditions at a near constant value despite changes in the environment and in the body by negative feedback mechanism. This is a longer question for 6 marks. It's asking about a camel. How is it able to maintain an internal temperature within this narrow range, even though the desert reaches temperatures up to 50 degrees Celsius? So first we have to say that thermoreceptors in the skin detect a change in the temperature away from the norm or the set point that acts as a stimulus. And then they send impulses to the hypothalamus or the thermoregulatory center in the hypothalamus. This sends impulses to effectors. The effectors are as follows. First, vasodilation, so more blood flows to the skin and more heat is lost by convection or radiation, and increased sweating so that the water can evaporate and the temperature will be lowered. And shivering is inhibited because shivering generates heat. We don't want to generate heat, we want to lose as much as we can. And decreased metabolic rate or respiration, so less energy is generated. And then you can say anything like panting or decreasing the adrenaline production. And then lastly, this isn't in the mark scheme, but it's in the book. So temperature goes back to normal and impulses from the thermoregulatory center stop. The definition of positive feedback: a system in which effectors work to increase an effect which has been triggered. For example, oxytocin, the hormone oxytocin, it is released when a child is being born. When the baby pushes against the cervix stretches. The stretch receptors in the cervix send impulses to the brain, and then the brain stimulates the pituitary gland to release the hormone oxytocin. And then oxytocin makes the uterus contract harder, pushing the baby against the cervix. So we're just increasing the oxytocin levels even more. And now a 3-mark question: compare between endocrine and the nervous system. The endocrine or hormonal system is slower, it has longer lasting effects, it uses chemicals transported by the blood, and it has a more widespread effects. Here, this is an epigenetics question from Unit 2. Explain how transcription factors cause the changes in the levels of some hormones in the bloodstream. So, some hormones are proteins, and genes need to be switched on before the hormones can be synthesized. And transcription factors bind to specific parts of the DNA so that RNA polymerase can bind, and then the certain hormone can be synthesized or can be stopped. And transcription factors can also switch off genes. We have 2 different types of hormones. We have steroid hormones, they're derived from lipids, and peptide hormones, which are made from proteins or amino acids. Okay, so they act in different ways. They both have the same effect. They act as a transcription factor. This means that they turn on or turn off certain sections of DNA. But the difference is, with peptide hormones, they release a secondary messenger. So they don't actually act as a transcription factor themselves, but they help in the release of a secondary messenger so that that secondary messenger can act as a transcription factor. This is because peptide hormones are not soluble in lipids, and as we know from Unit 1 or 2, I don't actually remember, that the cell membrane is a phospholipid bilayer. It's lipid, and only lipid soluble molecules can pass through it. And since peptides are not soluble in lipids, they cannot cross the cell membrane and actually cause the effect themselves. An example for this is adrenaline, ADH hormone, and insulin and glucagon. So, on the cell membrane, there are certain receptors that the hormone can bind to. After this binding happens, this will cause a series of membrane-bound reactions. This will result in a formation of a secondary messenger in the cell. An example for this, and the most popular example, is cyclic AMP, which is made from ATP. The secondary messenger will then activate a number of different enzymes in the cell to alter the metabolism. And the secondary messenger, cyclic AMP, will bind to other chemicals which pass into the nucleus and activate DNA transcription factors. But with steroid hormones, they are lipids, so they can pass through the cell membrane, so they act as a transcription factors themselves. An example for this is estrogen and testosterone. So they can pass through the phospholipid bilayer, and then they bind to receptors inside the cell, and then they form a hormone receptor complex and pass through the nuclear pore into the nucleus, and then act as a transcription factor. And then you have to say this for both of them. It regulates gene expression and the whole epigenetics thing. Here is an actual diagram showing you what's going on for peptide hormone and steroid hormone. The steroid hormone can actually enter because it's lipid, derived from a lipid, and then it binds to a receptor and it forms this complex. But with the peptide hormone, from the outside, it binds to the receptor, membrane receptor, and then it causes the release of a secondary messenger which will affect the metabolism of the cell. This is an example of how they ask about this topic in past papers. They ask about estrogen here, and they tell you that it is a steroid hormone found in animals. So you have to remember what's the difference between steroid and peptide hormones. So steroid hormones are the ones that are soluble in lipids, so they can enter and act as a transcription factor themselves. So it moves through the cell membrane, attaches to a specific receptor to form a complex, and then act as a transcription factor. And then how a transcription factor works: it binds to a promoter region of DNA, allows RNA polymerase to bind, so certain genes are expressed, and there is production of certain proteins. This is for the specific question. The proteins were involved in cell division because this is about breast cancer. The second part of 7C is about excretion and osmoregulation. Osmoregulation is keeping a constant osmotic pressure in the fluid of an organism. Simply, we don't want the blood and the cells to be too watery or too dehydrated. How we do this is, first, in the liver, urea is formed. Urea is made from deamination. Deamination is the name of the process. It's basically breaking down amino acids we do not need by removing the amine group from them. That amine group is removed to form ammonia. That ammonia combines with carbon dioxide in the urea cycle. And then amino acids cannot be stored in the blood because they're alkaline, so they would increase the pH of the blood because they have an amine group. So they're removed from the bloodstream by ultrafiltration in the kidney. So that's the first role of the kidney, ultrafiltration. Ultrafiltration occurs to the very high blood pressure in the glomerulus right here. This part of the kidney, the Bowman's capsule and the glomerulus is here in the cortex, while the loops of Henle are found in the medulla. Ultrafiltration occurs to the very high blood pressure in those blood vessels right here. This is done by the afferent arteriole. It keeps doing this. The afferent arteriole, which is this one here, the one going in is thicker than the efferent arteriole, the thinner one that's going towards the loop of Henle. This creates a high pressure. You have to know that between the Bowman's capsule and the glomerulus, there is the basement membrane. And you have to know the properties of the basement membrane. I'll discuss them in a moment. After ultrafiltration, where the blood is squeezed under very high pressure, and certain substances pass through the pores in the capillary walls. Only blood cells and large plasma proteins cannot pass through. The basement membrane acts as a sort of mesh. Then secondly, we have selective reabsorption. So while the filtrate travels around the nephron, certain ions and water are reabsorbed into the blood, because if we just lost all the ions and all the water, then our body won't get enough ions or water. This happens mainly in the proximal convoluted tubules. In the PCT, or proximal convoluted tubules, all of the glucose is reabsorbed. The PCT is covered in microvilli to increase the surface area for absorption of sodium ions and glucose. It reabsorbs 80% of the filtrate. Glucose is co-transported with sodium ions. You have to know this. And then lastly, the loop of Henle acts as a countercurrent multiplier. Countercurrent because the fluid moves in opposite directions in the two loops of Henle, as you can see here. This ascending and descending loop. And then it's a multiplier because it increases the ion concentration of interstitial fluid in the medulla, so that we have a more concentrated urine and we lose less water. It creates concentrated urine by the descending limb having narrow, thin walls and highly permeable to water, and the ascending loop having wider, thicker walls impermeable to water, and it also has protein pumps. The first thing that happens to the blood coming through from the afferent arteriole, the bigger one, it's ultrafiltrated. Ultrafiltration occurs here between the glomerulus and the Bowman's capsule. So this is the first step. So there is a very high blood pressure in the glomerulus, which is those little arterioles, because the afferent, the one that's coming in, is so much bigger than the efferent, which is going out of here. Okay, this forces all the small molecules out, but keeps the larger molecules like plasma proteins and the actual blood molecules. Okay, and then we have the selective reabsorption, which happens in the PCT, or proximal convoluted tubule, which is here. So what happens is that normally there would be too much water and ion loss, so the substances are selectively reabsorbed back into the blood. 80% of the filtrate is reabsorbed, so all of the glucose is reabsorbed, and some ions, and most of the water. And then here we want to create a concentrated urine, because if the urine isn't concentrated, we will lose too much water. This happens here in the countercurrent multiplier, which is the loop of Henle. It's called countercurrent because the filtrate moves in opposite directions in the ascending and descending limb. So, and it's called a multiplier because it makes concentrated urine. It multiplies the concentration. The descending limb is very narrow and it has thin walls and is highly permeable to water, so water can leave. But the ascending limb is wider and thicker and has walls impermeable to water. Let's explain this part here. As we can see here, the loop that's going upwards is going to be called the ascending limb. The ascending limb, as you can see here, is thicker than the descending limb, which is going down. Okay, so here we have the filtrate that just came out of the blood. It's coming first through the descending limb. The descending limb is thinner and it's permeable to water. So water, as you can see those blue little arrows, water is going to leave through the descending limb. But how are we ensuring that the water is gonna go out and not in? We want more water to go out, because when the water goes out of the descending limb, it's gonna enter the blood right here, so we can maintain an osmotic balance in the body, so we lose less water, because anything that's gonna stay inside here is gonna go to the urine if it's not collected in the collecting duct. Okay, so we know that here it's permeable to water and it's letting water out. How we're ensuring this happens is through the ascending limb. The ascending limb is going upward, the thicker one. It's impermeable to water, but it has certain pumps that pump out ions. Why does this help us? Because if you have lots of ions here, as we know from osmosis, the place with more ions, like Na ions and chloride ions, water is gonna go there. So we're taking all the ions to the interstitial fluid so that water can go there and follow the ions. The amount of water we drink, or how much we sweat, or how much protein we eat, and so the amount of amino acids we have in the body depends from day to day. So our body also has to osmoregulate, homeostasis. We need to keep the osmotic balance at a certain level. So how does this happen? This happens by the ADH hormone. It's a hormone that binds to receptors on the cell membrane of the epithelial cells of the DCT and the collecting duct. So here, the same diagram again, the collecting duct and the DCT, distal convoluted tubule. When the ADH binds to the receptors, the vesicles containing aquaporins fuse with the cell membrane. Aquaporins are protein channels for water, so this reduces the amount of water passed out in the urine. Basically, it makes the collecting duct more permeable to water. So they're more permeable to water. Imagine here, this is the collecting duct, and it has little channels of water. When the ADH arrives, those channels open, so the water can leave and enter the blood. Okay, I should probably go to red for this. So the water leaves the collecting duct and the DCT into the blood, so there is more water in the blood. This diagram really clears it out. It just tells you when there's too little water in the blood. For example, you sweat a lot, or you just didn't drink enough water that day, or you ate a meal with a lot of protein. This is what is gonna happen. First, the brain detects the water level, and then the pituitary gland releases the ADH. More water is reabsorbed by the kidneys, or this is from a lower level. This is for IGCSE, so you're probably not supposed to memorize this little details, but you have to know that more water is reabsorbed because the collecting duct is more permeable to water, so less water is lost in the urine, and then the water levels return to normal. Here is a very important past paper question. It asks how the loop of Henle acts as a countercurrent multiplier. As we said, countercurrent because the descending and ascending loop that's going up, descending loop that's going down. Just think of it as like descending, like descended from the sky or something. Okay, descending going down, ascending going up. They flow in different directions, so it's countercurrent. Multiplier because it's multiplying the concentration of the urine. So the fluid or filtrate flows in opposite directions, so countercurrent, and the sodium potassium chloride ions move out of the ascending limb, as we said before, so that there is more ions outside, and then ions move by active transport, resulting in decrease in water potential of surrounding fluid, and then water leaves the descending limb because it's the one that's permeable to water. The water can actually leave it. A question about glucose reabsorption for 3 marks. It occurs in the PCT, proximal convoluted tubule, using active transport in the membrane of the nephron, co-transport of glucose with sodium ions, and then it diffuses into the blood capillaries down the concentration gradient. How ultrafiltration removes urea from the blood. It's forced out by the high pressure of the blood through the pores in the basement membrane. We have lots and lots and lots of multiple choice question about this topic, especially about the kidney. Here it asks us which of the following produces ADH. So you have to know that the ADH is actually produced by the hypothalamus and then it's secreted by the pituitary gland. Okay, what does ADH act on? The collecting ducts and the DCT. The control of plasma concentration and blood volume is an example of negative feedback homeostasis. Osmoreceptors are the ones that detect the change in the plasma concentration. Another question from, actually, I don't know which year this is, but the kidney is involved in the process of homeostasis. How many of these parts respond to ADH? This is basically the same thing as this. Collecting duct, so just one. Here for 3 marks, it told you to draw a Bowman's capsule and label how many? Two parts of the graph. Okay, so there was a mark for this and then a mark for 3 labelings. Personally, I'd write basement membrane and efferent arteriole, especially because it already said efferent arteriole, so this must be the other one, even if you forgot, like, which is which. What else? More multiple choice questions. Here it asks which substances are normally filtered from the blood in the Bowman's capsule. So first, you wanna have hemoglobin filtered out because it's in the red blood cells and they're too big to be filtered. And then you also wanna have amylose, and you wanna have prothrombin because it's too big, so you're only left with glucose and sodium chloride. Which row shows the root? So you just basically have to know how it looks like, the nephron, and what are the labels. So we know that first we have the proximal tubule. The collecting duct is the last thing, so this is definitely wrong. The loop of Henle is not before the proximal tubule, so those 2 answers are eliminated. So PCT, and then we have the loop of Henle first before the distal tubule, and then the collecting duct last. Urea is produced by the metabolic breakdown of some organic molecules. It's produced urea. This is saying urea, not urine, so you have to pay very close attention. It's in the liver from excess amino acids. The last set of questions here, the diagram shows a single nephron. Which part of the kidney, W and X, are located? W and X is this, the Bowman's capsule and the glomerulus, and the PCT. So we already said that it is in the cortex. In which structure does ultrafiltration take place? In W, because it's the glomerulus. Which structures form part of the loop of Henle? So Y only. This is the descending limb because it's going downward. And we are done with 7C.